much secretion is happening.
Does the secretion or reabsorption of bicarbonate ions matter as well?
That will definitely affect it, but we need to talk about buffers to really understand that. But yeah, it will affect it also. Yeah. Okay. So again, the biggest problem is versus our blood being too acidic versus it being too basic. That is usually our problem. Usually we don't have the problem with our blood being too basic. So when we exercise a lot, lactic acid builds up. That's going to cause an increase of secretion of hydrogen ions into DCT. If you're in starvation mode, or if you have diabetes mellitus, where you can't get that glucose into your cells, so your cells think that you're starving. It thinks you haven't eaten anything. Either of these situations will cause what's called ketoacidosis. And what happens is, if you're in starvation mode, or if your cells can't get access to glucose, then your body, particularly your liver, is going to start taking your fat tissue and breaking that down and making glucose so that your cells can have it. And you can also start breaking down amino acids and then turning those amino acids into glucose so the cells can have it. When you break down particularly amino acids, proteins, sometimes fats, you produce a byproduct called ketones. And ketones are acidic. They make your blood become acidic. So this is something you have to be very careful about. People that go on the Atkins diet. Atkins diet is where you eat very little carbohydrates or no carbohydrates. You're eating high amounts of proteins and then decent amount of fats. The way that you know that you're doing it right, that you're in the zone of the Atkins, is you actually test your urine and you make sure that your urine is very acidic, because you want to make sure that your body is producing ketones, and then that means you have a lot of hydrogen ions in your blood, which means those hydrogen ions are going to end up in your urine. Now, you can do this for short periods of time, but they usually tell you with the Atkins diet, do it, get in that zone where you're in ketoacidosis by checking your urine. Also, you can smell a like a floral scent to your breath, and that's because of these ketones that are being produced. But anyway, you can stay in it, I can't remember, a couple weeks or something like that, and then you come off of it to let your body kind of recover in a sense, and then you can go back on it. So it is a very effective way of breaking down adipose tissue, but you have to be careful that you are, that if you're going to do the Atkins diet, you need to be overweight so you have that adipose tissue to break down. You don't want to be breaking down your muscles. So somebody that was on the thinner side obviously wouldn't do that. It just would make no sense because you start to waste away your muscles. But it is very effective to break down fat. Problem is that it's very hard to maintain, and it's dangerous to maintain something like that long term because of ketone acidosis. Now, this statement right here, don't worry about this. All this is saying is, if you have sometimes if you have high levels of aldosterone, instead of secreting potassium ions, the hydrogen ions will come in and attach to that receptor and take the place of potassiums. So if you have elevated levels of aldosterone for long periods of time, instead of secreting potassium like you should be, sometimes you'll start secreting too many hydrogen ions. And that can cause you to become too alkaline. Your blood can become too alkaline. But this, again, is a weird situation. This would not normally happen. It's if you're have really high levels of aldosterone for long periods of time. Okay. Now, we talked about water and solutes are regulated by aldosterone. and by ADH. Okay, so we talked about that, and that's important. That's why I'm mentioning it again. It's also important to know that ANP and BNP are opposing the effects of aldosterone and ADH. And again, we've already talked about this. I just want to emphasize that is important. If ADH and aldosterone, they work to increase blood volume and increase blood pressure, ANP and BNP do the opposite. They work to decrease blood volume and decrease blood pressure. Right? That's important. Don't worry about this part right here. So we talked about pH already, so we're good with that. Let me just mention on this slide, this kind of goes to what John mentioned. I really simplified things right here in terms of pH. And this is what I want you to know, this part of the pH. There is a little more that goes into it. It's not just, are you secreting hydrogen ions or not? It also has to do with when you secrete hydrogen ions, you reabsorb bicarbonate ions. And I'm not going to ask you anything about that. We just don't have time to go into that mechanism. So don't worry about the bicarbonate ion issue here. If you're interested, we can talk about it separately. Key thing I want you to know is regulation of blood pH due to increasing or decreasing secretion of hydrogen ions. Yeah, Jude.
Oh, no.
Okay. Let me just see. Okay, we talked about this already. Okay. We talked about this already. This is ADH. Okay. So last thing to mention is now that we've produced this urine, how do we get it out of the body? Right? So this is a nice slide here, nice image that shows the ureters coming out of the kidneys. The urine would travel through these ureters and go into the bladder. And again, that urine's going to be stored in the bladder until we have a reflex kick in and we get a sensation that we need to go to the bathroom. And then very similar to the way defecation works, if we want to go to the bathroom at that point, we're going to cause our one sphincter to relax, and then we're going to avoid urine goes through the urethra and out the body. Now, that reflex is called the micturition reflex. And again, it's basically like what is happening with defecation. So what we have here, when you look at the bladder obviously is a hollow organ, and in the wall of the bladder is going to be smooth muscle cells. And there are also a variety of receptors in there, stretch receptors. Now, at the base of the bladder, where the urethra comes out, right at the very top of the urethra, there are urethral sphincters. And again, just like what we saw in the anus, there's an internal urethral sphincter that is composed of smooth muscle. And then there's an external urethral sphincter that's composed of skeletal muscle. And we have control over the external skeletal muscle sphincter, and we don't have control over the internal sphincter that's composed of smooth muscle. And both of those sphincters are normally closed. They're normally contracted. And that keeps any urine that comes down into the bladder, because urine's constantly just drip, 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 coming from the kidneys, right? If we're doing, this is gone, 125 mils a minute, that's the filtrate we're producing. Obviously, we're reabsorbing most of that, but we just constantly have little drips of urine coming down the ureter and into the bladder. And once we get about around 200 mils or so in the bladder, and that will vary from person to person. Obviously, females seem to be more sensitive. A pregnant female is going to be really sensitive to this. But once you get about 200 mils in the bladder, And that's going to activate those stretch receptors. And that information is going to be sent up to the spinal cord here. And we're going to process that information in the spinal cord. At this point, you have no awareness of what's going on, right? Coming out of the spinal cord, we're going to have nerves that come down and cause that smooth muscle in the bladder to contract. And that's going to set up a high pressure here in the bladder. And then you're also going to have parasympathetic nerves that come down and cause that internal urethral sphincter to relax. Now, that's all happening without our awareness. Now, at the same time those things are occurring, the information is traveling along another pathway up to the brain, up to the cortex, obviously synapsing first in the thalamus because all sensory information except olfaction stops at the thalamus. Then it goes up to the cerebrum, and we have awareness of it. And if we want to void at that particular time, then we're going to send, or we're going to stop those descending voluntary motor pathways to the external urethral sphincter, cause it to relax. And the fact that the pressure is high here in the bladder because the smooth muscle is contracting, the urine will move from an area of high pressure to an area of low pressure out the body. Now, you can't just open the valves and the urine will come out. Some of it will, but that urine is going from this nice big roomy bladder into this little tube. So that's why you've got to contract the bladder to really get the pressure differences great enough that boom, it comes out, right? And obviously, if you cause accessory muscles to contract, you can force it to come out faster, right? Force it to come out faster. But if you decide not to respond to that stimuli, then that reflex will basically stop. And once you get, around another 100 or so mils in there, it'll fire off that reflex again. And you can keep doing that and keep ignoring that response until it gets to about 500 mils. And at that point, the pressure is going to be so high in the bladder from the smooth muscle contracting and so much urine in there that it's going to force both of those, it's going to force that external valve open and urine's going to come out whether you want it to or not. Now, one thing to just mention, you really should not ignore these reflexes, whether it's defecation or the bladder, because if you ignore them enough, you start to train your body to basically dampen those reflexes so that you don't respond to them as easily. And that's not good. It's not good for the sphincters. Over time, that can put too much pressure on those sphincters and cause them to weaken prematurely. And then when you get older, you have difficulty not urinating, or you have difficulty with incontinence, I should say. So you really should be careful about doing that. Okay, I think that covers everything. You guys have any questions? Okay, so I'll see you upstairs in about 15 minutes. If you want to take... Oh, let me say one thing. I'm sorry. Let me do one thing real quick. This has to do with your urinary histology. Just give me one second. Okay, so this is a slide that's on on our website, and this is a kidney slide. And what I want you guys to get from the kidney slide is I want you to be able to see corpuscles versus tubule. And these are corpuscles right here. Boom, 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 boom. And in the corpuscle, I want you to be able to identify the different parts. So this mass in here, that's your glomerulus, that big mass in there. The clear area, that's the space where the filtrate is going. And then that ring, it looks like a darkened ring of cells around there. And if you would magnify that, you'd see that they are simple squamish epithelial cells. That's your Bowman's capsule. So if I show you that, if I put the pointer here, you should know that's the glomerulus. If I put the pointer here, you should know that's Bowman's capsule. If I put the pointer there, you should know that that's the space. You should know the whole thing is called the corpuscle. And then what you're looking all around it, all of these rings here. And again, if you magnify this, you'll see that those are rings of simple cuboidal epithelia. Those are rings of PCT or DCTs, just cross sections of PCT or DCT. And I'm not going to ask you to identify PCT versus DCT. It's a little bit tricky, and I'm just not going to ask you to do it. But you should know that this is the corpuscle, and then out here are PCTs and DCTs. And where you see a bunch of corpuscles, then you should know that that would be the cortex area of the kidney, right? Because it's in the cortex where you find the corpuscle and the PCT and the loop of Henle and the DCT and collecting ducts. So up here where the corpuscle is and the PCT and the DCTs, that's the cortex region. And if you would go deeper into the slide, then you'd start seeing these loops of Henle's. And they generally look like long, kind of cross-sectional rings. Sometimes they're small, it just depends. But that's all I want you to know from the kidney. You should be able to look at that and know that it is a slide of the kidney. You should be able to identify corpuscles and the different parts of those corpuscles. Okay, now I'll see you guys upstairs in 15 minutes.